Domain 7 is research integrity. Little bit of background. Um, if the authors did not adhere to the principles of research integrity, these results can be unreliable even if the study experimental design and analysis were reliable. Lack of research integrity can decrease the confidence in the study findings. What's the purpose? Ensure that the authors adhere to the principles of research integrity. Okay, the first signaling question 7.1 is, did the research team obtain ethical approvals and any other regulatory approvals required to perform the research prior to the start of the study? Now, animal research should be performed ethically in a humane way. While university ethics boards can confirm the existence of ethical approval, additional licensing requirements, for example, home office licenses in UK, may be necessary before the research can be conducted. This is to ensure that the principles of replacement, that is, methods which avoid or replace the use of animals, reduction, methods which minimize the number of animals used per experiment, and refinement, methods which minimize animal suffering and improve welfare, are followed during scientific research. In some countries like the UK, preclinical studies conducted to justify human clinical trials are required to follow good lab practice regulations. So there has to be it has to be done in the appropriate way with all the regulatory approvals. Okay, classify this as yes or probably yes if the study authors have provided details of ethics approval or any other regulatory approvals and standards that they used in their research. Classify this as no information if the study authors did not report the details of ethics approval, other regulatory approvals and standards followed. Classify this as no or probably no if it was clear that the authors did not obtain ethics approval or other regulatory approvals as relevant, or have clearly deviated from the license that was obtained. Okay, before we go to the signaling question 7.2, we need to understand what data corruption and data cleaning means. In this context, data corruption means unintentional human errors when handling data. And that has the potential to affect the quality of the study results. And it could be a possible reason for lack of reproducibility because they cause misclassification of exposure or outcome. So if the data gets corrupted inadvertently, then you won't get the correct results. Data cleaning is the process of identifying and correcting these errors, or at least attempting to minimize the impact on study results. And there are different methods used for data cleaning, and the methods used for data cleaning can have a significant impact on the results. So the signaling question 7.2 asks, did the authors take steps to prevent unintentional changes to data? Now, the best way to minimize data corruption and decrease the impact of data cleaning is to avoid data errors in the first place. While there are many data handling guidelines about the protection of personal data, there is currently no guidance on the best method to avoid data corruption. The UK Digital Curation Centre provides expert advice and practical help to research organizations wanting to store, manage, protect, and share digital research data. Maintenance of lab logs, accuracy measures between lab logs and data used, and use of password protected data files can all decrease the risks of unintentional changes to data. Okay, how are we going to classify this? Classify this as yes or probably yes if the authors took steps to prevent unintentional changes to data. For example, a password protected sheet or log to indicate any changes in data. Classify this as no information if the authors did not report the steps to prevent unintentional changes to data. Classify this as no or probably no if the steps taken by the author were clearly inadequate to prevent unintentional changes to data.